Okay, the main concern of our discussion today, class, is all about the components. The other term for this one is the organic compounds that is present in our human anatomy, which we call as macromolecules. Macromolecules means large molecules that is present in certain entity, may it be a human being or may it be an animal. For as long as they are large molecules that is composed of covalent bonds between the main elements carbon, hydrogen, oxygen, and nitrogen, or we can shorten it as CHON. In your module, it is CHON for protein, but for and also for nucleic acids, but for carbohydrates, it's just CHO, which means there's no nitrogen present in carbohydrates. And these four elements are the main components of the macromolecules that we have in our human anatomy. And they're bonded together with a so-called covalent bond, since all of these are nonmetals. And they function differently. The four macromolecules that is present in our human anatomy are as follows. We have carbohydrates, lipids, proteins, and nucleic acids. Each of them functions separately. Each of them have distinct features that is allowing a human being or a living creature to perform its biological function or to perform its chemical function. So we have here macromolecules are composed of polymers, but each of the polymers are composed of a single monomer, as an example of which carbohydrates is composed of a long chain of sugar. We call these polymers, but when we talk about the monomer of carbohydrates, the monomer of carbohydrates is said to be simple sugar or saccharide. So mono from the term mono means one, which means the building block, the single building block of carbohydrates. So for example, in proteins, the monomer of proteins is a peptide bond of amino acids, no? And the polymer of proteins is a polypeptide chain. It's composed of one or more or two or more peptide bonds. So again, the polymer is a long chain of molecules composed of the building blocks or the monomers of the said macromolecules. Okay. Um, for lipids, which is not stated here, the monomer of lipids is glycerol. Yes, glycerol. It's a syrupy substance, um, somehow sweet in tasting, and it is composed of three fatty acid chains. When we combine monomers class in the process known as dehydration, we will create polymers. We can also break the polymers into monomers in the process known as hydrolysis. Dehydration means um, removing water in each of the monomers. Hydrolysis means adding water to the polymer. As such, the polymer will be broken down into monomers. So in your frame right now, the synthesis and the breakdown of polymers follows the two processes. The first process is dehydration. Dehydration means two monomers will be bonded together. Um, in this case, the two monomers will be linked with a so-called hydro, hydrolysic reaction or hydrolysic chain, and they will be bonded by removing the water in these monomers. There's an example of the illustration in your module. Sorry to mention, it's not hydrolysic, it's hydrosidic linkage or hydrosidic um, bond between two monomers of carbohydrates. You have there the glucose and fructose. If you dehydrate, or I mean, if the two monomers will participate in the dehydration reaction while removing water, if you notice the ones with the white circle, if you remove the hydroxyl group of glucose and the hydrogen of fructose, which means that there are two molecules of hydrogen and one molecule of oxygen, which is water. And then the oxygen will, of course, follow the octet rule. So it will have two bonds. The bond between fructose itself and the bond while losing hydrogen will be bonded to the glucose. That's the glycosidic linkage to create the polymer of the two monomers, fructose and glucose. And the polymer for combining glucose and fructose is sucrose. Also, class, if you are going to, good afternoon, Miguel, good afternoon. Also, class, when you're going to reverse the process, the from a polymer, it will be disassembled or will be broken down into monomers from sucrose. If you wish to remove the two, or if you wish to separate the two monomers from sucrose, which is fructose and glucose, you will undergo the so-called hydrolysis reaction. It's a reaction that is the reverse of dehydration, and it literally means to hydrolyze or to add water. But it's not as simple as putting water to it and then mixing. Still, it's in the process of chemical reaction. So again, the two processes that involves in breaking down and synthesizing polymers are as follows: dehydration and hydrolysis. So here is an example of uh, monomer, short monomer, uh, with the blue link H and OH. You remove the OH from one monomer and then the H from another, it will result to H2O, which is water. That's the dehydration part, removing water from each monomer. As a result, you have a longer polymer from the result of the linkage between the short polymer and the unlinked monomer. Letter B is hydrolysis. So from the long polymer, it is now adding a water molecule to break the bond and thus removing the linkage between the third and the fourth carbon, which will end up with um, COH is alcohol, right? So the one on the right, and then also the one on the left is also another alcohol, which is OH on its ends. The one, two, three, and four are carbon chain. The backbone of the protoplasm of life or the macromolecules is carbon. The backbone of the chain between proteins, carbohydrates, lipids, and nucleic acids are carbon. And the one, two, three, four there are what we call as the carbon chain class. So this is an enlarged picture of letter A from the picture before. Dehydration reaction, which means you synthesize a polymer from two different monomers. This is really removing water from the molecule itself, and the new bond is created as we consider it as the longer polymer or the chain of carbohydrates, shall we say, if this is sugar monomer. What are the different monomers of sugar, or what are the different monomers of carbohydrates? There are five or six of them: glucose, fructose, galactose, you have ribose, and deoxyribose, alimaride. So these are the five monomers of um, carbohydrates. The ribose and the deoxyribose is an important monomer as well for the DNA and the RNA, or the distinctive monomer sugar for DNA and RNA. This is the picture for hydrolysis, or the breaking of the polymer into two different monomers by adding water to it. You can also associate this as a sugar. You place it into water, and then the sugar molecules will be disintegrated. No, it's like the sugar melts from the water by the presence of water. Each of the thousands of macromolecules class have variations of monomers, and these monomers are still can be added or can be combined into different polymers, or it can be broken down into simple monomers. As again, you have to use the processes dehydration and hydrolysis. Please do not interchange the two terms. I hope I placed that in your module. I think that's not found in your module, no? So please make some notes about it, class. So let's start with the first macromolecule known as carbohydrates. The polymers of carbohydrates is sugar. The monomer of carbohydrates is saccharides or simple sugar. Uh, and it is the monosaccharide if there is one monomer of sugar. And the monosaccharides are as follows: glucose, fructose, galactose, ribose, and deoxyribose. These are the five monosaccharides. But if you combine two monosaccharides, it will end up with disaccharides. And if two or more than two, it will be polysaccharide already. So carbohydrates are macromolecules that are composed of polymers, which is called polysaccharides or many sugar building blocks. For the monomers of carbohydrates, we term it as monosaccharide, and then the empirical formula. Shall we say the empirical formula for monosaccharide is CH2O. The most common of these monosaccharide is glucose with the molecular formula of C6H12O6. And these monosaccharides are classified in the location of their carbonyl and the um, carbonyl connected with ketone or connected with an alcohol or aldehyde rather. Carbonyl, aldehyde and glucose will make it aldose. Aldehyde, ketone plus glucose makes it a ketose. But this is more like chemistry two. Did we have this chemistry two? Naming organic compounds. So probably this might not be one of the things that we need to memorize in this lesson because our main concern here is all about the information FYIs of carbohydrates. Let's leave general chemistry to discuss these molecules class. So we have a three carbon sugar known as triose with the addition of an aldehyde. Aldehyde is the carbonyl bond, C double bond to O. The hydroxyl is replaced with a carbonyl. Notice the one with the highlight. That is an aldehyde. So the glucose plus an aldehyde makes it a glyceraldehyde or an aldose. And then for ketone is C double bond O, but that is in the without the bond for hydrogen. The difference between the aldehyde is the bond of the carbonyl to a hydrogen. Our clue there before grade 11 was CHO for aldehyde. And for ketone, it's C double bond O. But never mind with this one class. No need to worry about this. Just an FYI. For a 5 carbon sugar, you have a ribose and a ribulose. No need to do as such, Miggy. The important thing there is, the molecular formula of three carbon chain. You have here the three carbon chain. What was it? C3H6O3, then 5 carbon sugar, C5H10O5, but the most common of which is the 6 carbon sugar, which is C6H12O6. So we have here the three uh, basic monosaccharides. That is the backbone of the disaccharides, glucose, galactose, and fructose. Together with ribose, deoxyribose, and a component for an contrary to ketose between ribose is the ribulose. Ribulose. So I think the ribulose makes it the sixth monomer of carbohydrates. Uh, as mentioned by Miggy, do we have to review aldehyde and ketone? No need to review that one, Miggy, and so as with others. It's just I made I maximized the data that is present in this um, presentation given to me by Pearson Incorporated. But with respect to memorizing aldehyde and ketone, no need to worry about it. Let's talk about disaccharides, the combination of two monosaccharides by the dehydration reaction and the covalent bond that exists between the two monomers of disaccharide is known as the glycosidic linkage. For example, we have here glucose and fructose. If we dehydrate glucose and fructose by removing water as one of the um, what's the term there that we use in order to differentiate the the bond of of benzene ring, di ba? Benzene ring is siya. Forgot the term that we used last time when we were grade 11. So if you notice class, the hydroxyl of the first carbon, shall we say, is the first carbon, and the hydroxyl of the other monomer, which is glucose, the same. We remove one of the hydrogen in the bond between the hydroxyl OH and then both hydroxyl in the first glucose. Thus, we liberate water from the glycosidic linkage. As such, when you combine two glucose units or two monomers of glucose, that will result in maltose. Where can you mostly find maltose class? Where can you mostly um, hear or associate maltose in the food that we mostly eat? Barley. Barley is high in maltose or malt sugar. And barley is the key component for beer. Which means that beer, if you will keep on drinking beer, it is high in sugar. Parang mahinay ka og beer, flexi, and again. The, the combination between glucose and fructose, if glucose and fructose participates in a dehydration reaction, it will synthesize sucrose. And sucrose can be seen in our kitchen, no? Most especially table sugar. The not medical, the technical term for sugar or table sugar is sucrose. Okay, so you can dehydrate or you can hydrolyze sucrose or sugar by putting it into a water, and if you notice, the sugar components will be dissolved in the water. Thus, that is the result of the hydrolysis reaction to remove the two monomers of sucrose into glucose and fructose. What about if I will to combine glucose and galactose class? Glucose and galactose. If you allow glucose and galactose to participate in a dehydration reaction, it will result to the sugar found in milk, which my digestive system cannot tolerate. Starts with letter L, that is lactose. No? Glucose and galactose to participate in the dehydration reaction will result to lactose. And lactose is a sugar found in milk. And I myself is lactose intolerant, which means my digestive system cannot tolerate too much lactose. Thus, it will give me problems later on. Either it will give me bowel movement or it will give me pain in my stomach. Polysaccharides are a combination of more than two monomers of sugar. Examples of which we have starch, which is the sugar storage found in plants. We also have um, chitin or chitin, the sugar storage found in some exoskeletons of arthropods. Chitin is spelled CHITIN. And also the sugar storage found in animals. Now, what is that sugar storage found in animals? The term there is glycogen. I think it's not found in your module, no? I hear it's in your module class. Um, Above the illustration of sucrose, these simple sugars, disaccharides, and polysaccharides, long chain of cellulose, chitin, and glycogen. So it's in your module class. Cellulose or starch is stored in the cell walls of your plant cell, which is eventually will become cellulose. These are the sugar stores found in plants. For animals, it's glycogen. And for the exoskeleton of some arthropods, we call it chitin or chitin. This chitin class is responsible for strengthening the thread used for dissection. Kanang thread nga itahi ni human opera. Are you familiar with that? Boys, I think you're familiar with it. The thread that is used after you are going to be circumcised. And these will just decompose after the wound will heal. This is made of chitin or chitin. And that is sugar or carbohydrates. Minya is going to join our call. Okay, so in plants class, the starch or the cellulose is mostly termed fiber. Um, if we associate fiber in the food that we eat, these are the grooms or the sweepers of our digestive system, and indeed they are. But just an FYI, limit yourselves from eating too much fiber in the evening because fiber is hard to digest. Between protein and carbohydrates is harder to digest. Huh? Probably you need to eat these fiber in the morning or in the afternoon, but not much in the evening. Good afternoon, Ninya. We have Ninya in the session right now. Thank you for joining our call, Ninya. So let's proceed then with some illustrations regarding the cellulose or starch found in plants. These are some. These are examples of scanning electron microscope results of a starch and glycogen found in the plant and animal cell respectively. This is the chloroplast with the starch granules. Glycogen is the sugar stored in animals. I did mention it a while ago. They can be mostly located in the liver and some muscle cells. Now, if you notice some of the meat that we tend to eat as raw meat, like raw fish, raw raw squid, kinilaw in short, they taste a little sweet at some point, no? With the addition of some sour taste by some additives like suka and other ingredients there. But the main meat, especially the meat itself of the fish, is somehow tasting like sweet. Somehow has a sweet taste in our taste buds. That's also the same when you're going to eat sushi. Have you tried eating sushi? Go to some um, Italycans in Cebu. There are a lot of sushi in there. Not much in Bohol, I think. Not too soon in Bohol. Okay, so we have here the structural polysaccharide in cellulose, which is the fiber, no? Which is the fiber found in plant leaf or plant fruits or plant flowers if they are edible. So let's focus our attention on the non-chemistry part, okay? Not much of this one. Okay, we have here chitin here. Chitin is another structural polysaccharide which is found in the exoskeleton of some arthropods. They provide structural support for the cells, for the cell walls of many fungi as well. So here is an example of chitin, remnant or um, hollow body of anong tawag dito? Parang gangis ba yan? They will be left there in a tree and they can be cultivated to be made as a thread. As well, I did mention a while ago that chitin is used to make a strong and flexible surgical thread that decomposes after the wound or incision heals. Um, this is not one insect on top of the other. This is an insect that goes out of its skeleton. Shall we term this metamorphosis? Probably this will metamorphose from an insect to a winged insect, no? From a crawling insect to a winged insect. I think this is a moth. No, it's not. Or it's not. So those are examples of carbohydrates, its monomers and polymers. So again, what is the monomer of carbohydrates? It's called simple sugars or monosaccharide. And then the examples of the monosaccharide or simple sugar include glucose, galactose, and fructose. Combining of which to participate in the dehydration reaction results to maltose, sucrose, and um, lactose. And the rest of these, if we combine to another dehydration reaction, it will result in polymers like chitin, um, starch, and glycogen. So to follow, we have here lipids. Probably in your module is the third one. It's one of the large biological molecules or large macromolecules, and these lipids class are the richest source of energy, shall I say. What do I mean by this richest source of energy? If we define carbohydrates class, carbohydrates is the immediate source of energy, the energy that we need on the spot. But if we run short of the sugar, if we run short of carbohydrates, then our body made use of the fats that that we have to synthesize these fats and turn it into energy in the form of ATP. No? If we are fond of eating sweets and sugar and we are not using this energy, these sugar molecules will be deposited in your human anatomy in the form of fats. Most of these sugar components will be stored in your belly, in your arms, especially for the girls. It's somewhere here, in the hips of most girls. But most of these really happens to be located in your belly, as you are not using this energy. No? Lipids as well are the main components of all cell membranes, as our cell membranes is made of phospholipid bilayer. There are good and bad lipids, or there are good and bad um, fats that we have in our human anatomy class. Some of these good will become bad if their concentration is already beyond minimum. No? Fats are constructed with two smaller molecules of glycerol and fatty acids. Glycerol is the monomer of lipids. But if this glycerol will be combined with one, two, three, four, and more fatty acids, then it will become glyceraldehyde or triglycerides rather. Glycerides, diglycerides, and triglycerides. That's like monosaccharides, disaccharides, and trisaccharides. No? So we have here some structures of glycerol with three fatty acid tails. We termed it as triacylglycerol, or it's also called triglyceride. Again, this is triglyceride or triacylglycerol. If you have two fatty acid tails, the yellow ones are the fatty acid tails. If you have two fatty acid tails, the name there is diglycerides or diacylglycerol here. Fats separate from water because they are not um, soluble in water. They're only soluble in uh, non-polar solvents. For example, ether, alcohol, and the likes. In fats, the three fatty acids are joined with the glycerol in an ester linkage, creating a triacyl glycerol, also known as triglyceride. So it's safe to say that the monomer of lipids is glycerol or glycerides. Okay. I think there's a definition of glycerol or glyceride in your module. No, it's still the same. A glycerol with three fatty acids is triglyceride or glyceralcyl triacylglycerol. Fats or lipids have a good and a bad components, saturated or unsaturated. When you talk about saturated fats class, these are the bad fats. When you talk about unsaturated, they are the good fats. Example of bad fats or saturated lard, no, or animal fats. They are saturated fats, and they are not that good with respect to your health, the health is concerned. But when you talk about unsaturated, a good example of this is oil. No? What is your what is your indicator that they are bad? What is your indicator that they are bad or they are good? If you look at animal fats class, if they are going to be placed at a lower temperature, what happens to these fats?
Hardened.
They clump, they harden. And
Freeze.
imagine if this will go to your blood vessels, they will accumulate in your blood vessels. In short, your blood vessels will be clogged, and if it will be clogged, you'll experience the condition known as atherosclerosis. And if you have that condition, then your blood pressure will increase because there's only a minute space that the blood can rush in your arteries. And if there's a high blood pressure, then complications will happen. If this clogging of the arteries is found in your heart, then you'll experience myocardial infarction, also known as worst case scenario, cardiac arrest. If these arteries will be, if the carotid artery will be blocked by these bad fats, this will in turn became a problem, and this problem will be called stroke or cerebrovascular accident. The importance or the relevance of which is that the blood carries the oxygen, and if this oxygen will not reach to the cells that need this oxygen, then the cells will die. For example, if your coronary artery delivers blood to your heart, and if this will be blocked, then the heart cells will not anymore receive the oxygen that it needs. That is why this term is called coronary heart failure or coronary heart disease, also known as cardiac arrest. Worst case scenario na ng cardiac arrest. Example lang, if daghan gyud og tambok nga kanon. But there are good fats that is needed in our body, like the unsaturated fats, an example of which is cholesterol. Cholesterol is an unsaturated fat, which is a good fat. Cholesterol is needed in our body to synthesize vitamin D. You cannot just get vitamin D from the rays of the sun. Let's say you go out at 6 a.m. in the morning and you bathe yourself in the sun and you say, "Give me vitamin D, Lord." So you'll bathe yourself in the sun. No, it's not. Without the presence of the cholesterol, this radiant energy that comes from the sun is just merely energy. But because of this cholesterol that we have in our body, the energy that is, or the radiant energy from the sun will be converted into calciferol, which is the technical term or the medical term for the so-called vitamin D. That is why it is calciferol because it's the combination of calcium as the element that strengthens our bones and teeth with the aid of the cholesterol, which tries to generate the vitamin itself, known as vitamin D, also known as calciferol. So if you will be asked, what is the name of vitamin D? Do not say calcium. Vitamin D is good for the bones, but vitamin D is not named calcium. Vitamin D is calciferol. So they are good. However, again, too much of everything is bad. For example, if you have too much intake of the good fats, then it will become already something not good. Like alcohol class. Alcohol is good in our human anatomy. We need alcohol. Our body functions needs alcohol. But then again, if too much of this alcohol will be taken in by an individual, then it will now affect the nervous system of that human being. Uh, it will affect the proper functioning of the nervous system. Thus, it will create hallucinations. Worst case scenario, it will generate unlikely behavior. No? So these are examples of good and bad fats. Do not just always consider that fats are always bad. It also has its good uh, effects class. As in the energy pyramid or the food pyramid, fats is in the topmost level. We still need fats, but of a minute concentration. Here is the saturated fats or oils, and this is the and this is the saturated fats rather, which is the animal fats or lard. It will harden at room temperature, but this is the unsaturated fat. This will not harden at room temperature. These are oils, palm oil, vegetable oil, coconut virgin oil, or the likes. We also have lipids that is responsible for other um, processes, chemical processes in our human anatomy class, which is also in relation to nucleic acids, and the other one is proteins. No, so later on we will talk about it. Fats have a medical term or technical term known as adipose tissue. Rich they also have its job, like they cushion the vital organs. Here, adipose tissue, they cushion vital organs, they insulate the body, they absorb heat, as our internal environment also performs mechanical processes, and it will release heat because of the fats. It will absorb the heat, and it will cushion some of the internal organs, prevent them from generating friction to each other, thus creating wounds. Fats in cells are termed phospholipids. Um, they are the ones that controls the entrance and the exit of elements or chemicals or molecules that goes in and out of your cell. We also have here this one class. Steroids are lipids with fuse foring. Steroids are good and at the same time bad. Steroids are needed by some pharmaceutical companies as a component of their medication. One of these medication that uses steroids are those that medicate problems in the kidneys. My dad is one of them who had steroidal intake of medication because my dad had problems in the kidney, and as a result, if you have an intake of steroids, at some point you bloat or you tend to be bigger because it absorbs much of the water as well. Uh, Lordsa as well is intaking some steroid medication. That's why if you are familiar with Lordsa Dano, Lordsa has a big bulky body because of the steroid intake that he had in his younghood stage. Cholesterol is an important steroid for animals as they are the main components for blood cardiovascular, the main components for synthesizing of vitamin D. However, if there's too much components of cholesterol in the blood vessels, then it will result to problems in the circulatory system, also known as atherosclerosis. So let's proceed with the third one. The third macromolecule is proteins. And proteins have a diverse function in your human anatomy. It is enumerated in your module class. We have enzymatic proteins, structural, storage, transport, hormonal, receptor, contractile, defensive. And I hope these are all mentioned in the presentation. But what are the monomers of proteins class? The monomers of protein is peptide bond. And what are the monomers of a peptide bond? If you are going to hydrolyze a peptide bond, it will result to the 20 basic amino acids. In short, the building blocks of proteins is amino acids. Okay? And and in your module class, in your activity, I have there activity one, give the name of the amino acid with given the abbreviation. Activity 2, give the abbreviation of the 20 amino acids. You can search it in the internet or I will give it to you right now. I think I did not include that in the module specifically because I wish you to search it and have it as notes. It may be integrated in your module or it may be just placed in your notebooks class. So here are the 20 basic amino acids that are considered building blocks of proteins. Maybe I will use um, Microsoft Word and I will write it there. I hope I can find our whiteboard. So we have here valine. I'll make it black. Valine, leucine, isoleucine, alanine, arginine, glutamine, lysine, aspartic acid, also known as aspartate, glutamic acid, glutamic acid, also known as glutamate. Then we have proline, cysteine. Where do you think is next to cysteine? 17, no? Threonine, methionine, histidine. Most of the students are really having a problem, or most of them commits a mistake in histidine. They always mention the term histamine. Histamine is an allergen, huh? It's not an amino acid. Then we have phenyl, alanine, tyrosine, tryptophan, asparagine, glycine, and serine. We will have a rediscussion of these molecules. Will have a rediscussion of these amino acids once we will discuss protein synthesis because the building blocks again of a protein is the 20 basic amino acids. So one of the indicator of how you're going to memorize the three abbreviation is the use of the first three letters. For example, valine is val, leucine is leu, isoleucine is ILE. Isoleucine is ILE. That's the three-letter abbreviation of isoleucine. Alanine is ALA, arginine is ARG, glutamine is GLN. There's a difference, huh? Glutamine is GLN, lysine is LYS, aspartate is ASP, glutamate is GLU, So there's the difference between glutamine and glutamate. Glutamine is GLN, glutamate or glutamic acid is GLU. Rulin is pro, cysteine is CYS, threonine is THR, methionine is MET, histidine is HIS, phenylalanine is PHE, tyrosine is TYR, tryptophan is TRP, asparagine is ASN, glycine is GLY, serine is SER. Okay, these are because if you use the three letters of asparagine as ASP, that is already the abbreviation of aspartate, which is ASP. We'll soon have a rediscussion again of these terminologies as soon as we discuss protein synthesis. So for the sake of answering your module, you please have this researched and have it written as notes in your um, ano class. Have it as a notes in your have it as notes. It can be integrated in your module or it can be written in your notebook. So again, proteins are considered to be structural, storage, transport, cellular movement, defense, and the like. So let's start with um, let's start with the first one, enzymatic proteins or enzymes. Proteins can be considered enzymes and they function to accelerate chemical reactions. Enzymes could either be a catalyst, which means they will not participate in the reaction, rather they are the igniters of the reaction, or they will participate in the reaction as enzymes itself. No? Example of which is a digestive enzyme. Storage proteins, examples of which is the protein found in eggs, which is albumin, the protein that is found in milk, which is casein. How about the protein found in skin that makes it elastic? That's elastin. Okay. But these are structural proteins already, but it's just for maybe it will not be mentioned later on. What about the protein that is found in your hair? That is keratin. But most of these are structural proteins. I think it will be mentioned later on. I am making it fast because it's almost time class. I hope you will understand. So we have hormonal proteins. Example of a hormonal protein is insulin. Insulin is a protein, and this hormone or this protein is responsible for the balance of blood sugar. No? Insulin reacts with the pancreas, or rather, insulin is secreted by the pancreas, and this will raise blood sugar level, or rather, reduce blood sugar level. If there is more blood sugar, if there is more sugar in the blood, then insulin is released so that the cells will absorb these sugars found in the blood, and thus it will normalize the blood sugar. If you have low blood sugar, then you will be then your pancreas will release a hormone, though it's not a protein. The hormone there is glucagon. Contractile proteins or motor proteins, examples of which we have the actin and the myosin for muscle contraction. We have the flagella for a sperm cell to contract or enable it to motility, and some cilia for some unicellular organisms, which allows them to move or which is responsible for locomotion. Defensive proteins are antibodies. Examples of these defensive proteins are antibodies, which tends to program a specific virus or bacteria, and this will be read already by your human anatomy. Thus, if your B cells, in particular in the lymphatic system, will read that there's a specific bacteria that attacks your body, then the B cells will release the T cells, which is the cells that targets that specific bacteria or viruses, and these are already immune to the said bacteria or viruses. Transport proteins, example of which is the hemoglobin that is in your blood, which transports oxygen to and from the lungs. In your cell membrane, you have different transport proteins. You also have the channel proteins, receptor proteins. Microtubules is an example of structural and motor proteins. So these are mostly found in the cell membranes, the transport proteins. Receptor proteins, they are found mostly in the synapse of the muscle junction, the synapse between sensory neuron to the muscle cell. No? And these receptor proteins receives the acetylcholine, ACTS, the one with the color red. They're the acetylcholine or the signaling molecules in a muscle contraction. Structural proteins, I did mention a while ago. Keratin for hair, horns, feathers, skins, appendages, and the likes. Collagen for or elastic. Collagen is for tendons class, and then elastin for skins to become elastic, such that it will prevent the tearing of the skin. Also for silk, silk fibers and cocoons, they are highly made of keratin. Here's a structural protein, microtubules and microfilaments. We have storage protein. This is Golgi and or rough ER. We have transport protein. This is found in facilitated diffusion. Receptor protein, contractile protein, microfilament, defensive protein. These are B cells class, specific B cells in your lymphatic system. Hormone receptor, part of the plasma membrane. Sensory protein, still part of the plasma membrane, but not part of the diffusion, facilitated diffusion. Then gene regulatory protein, that's your DNA, and the likes. Enzymes can be catalysts that speeds the reactions, or uh, take note that catalysts are speeding up the reaction, but they are not part of the reaction. Example of which is chlorophyll. Chlorophyll is a catalyst in photosynthesis, but chlorophyll will not participate photosynthesis. The participants of photosynthesis is light, water, and carbon dioxide. Enzyme, example of an enzyme, um, amylase, salivary amylase is an enzyme. Here, amino acids are composed of carbonyl and hydroxyl. I think this is found in your module class, the structure of an amino acid. Let's proceed with the last because it's already time. Last one. Nucleic acid is just composed of two, the DNA and the RNA, the difference of which is the sugar found in it. We have the deoxyribose and the ribose for DNA and RNA respectively. The ones that I skip are the structures of your protein. No? Um, I'll just share the presentation in your Edmodo so you can review the PowerPoint class. Okay, here we have nucleic acids and the polymers of nucleic acid. The building blocks of nucleic acid is nucleotides. The two types of nucleic acids are the DNA and RNA. DNA is responsible for the genetic blueprint of our life, whereas the RNA is the slave of the DNA, which means if what is the blueprint found in the DNA, the RNA transcribes it in the process known as protein synthesis. And then again, the structure and the importance of these two nucleic acids will be repeated in our discussion um, for protein synthesis. It is not mentioned in your module, but there is also another example of nucleic acids, the CAMP and the cyclic adenosine monophosphate. These are they are not nucleotides, but they are term there is phosphates found, which is a subcomponent of nucleic acid because nucleic acid is composed of phosphate, sugar, and um, nitrogenous bases. The four nitrogenous bases there we have adenine, thymine, guanine, and cytosine, and but in the RNA you have the uracil. And then again, we will have this rediscussed as soon as we have the discussion on protein synthesis. The the summary of nucleic acids class is found in your second page of your module. Please review that part because some of the questions might come out there, like what is a nitrogenous base that is present in RNA which is not found in the DNA? The answer there is uracil and the likes. Okay, so I hope that you learned something for today. After this, you may start answering your module. You may search the answers in the internet, or if not, you can just make use of the module as your main source of information class. That would be all for today. Goodbye, and thank you for your attendance.
Goodbye
Goodbye
and
and
thank
thank
you,
you,
Sir
Sir
Jet.
Jet.
Bye bye.